Hello everyone. My name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences in Bioengineering, IIT Kanpur. Today, we will start with the chapter Photosynthesis in high plants. So, this is the first lecture and we'll basically study about the introduction and the basic experiments that were performed at that time to prove that what are the components that are important for photosynthesis. So, the first topic will be what is photosynthesis? So, as the name suggests, it contains the term photo, right? It has this term photo. So, what does photo means? It means light. So, we all know that a photosynthesis is a process which is used by the plants, right? And what does it do? So, the plants. So, what is photosynthesis? It is a process. It is a process by which plants make their food. Plants make their food. This thing we all know. And what do they require to make their food? So they require this to make their food. Food is light. So the photosynthesis is a process by which plants make their food in the presence of light. Right? In the presence of light. And because of this reason, it is named as photosynthesis. That is synthesis in the presence of light. Okay. Now, where does photosynthesis take place in the plant? So the photosynthesis takes place. It takes place in the leaves. Okay. It takes place in the leaves. So leaves are the part of the plant or the tree that synthesize the food for the whole plant. Right. Now, what kind of reaction is the photosynthesis? So, the photosynthesis is what? It is enzymatic reaction. It is enzymatic reaction, meaning it involves a set of enzymes to complete this synthesis, right? And hence it is known as enzymatic reaction. Fourthly, it is also known as physicochemical reaction. So, it is also known as photosynthesis is also known as physicochemical reaction. Now, why it is called as physicochemical reaction or what is physicochemical reaction? So, the physicochemical consists of two terms, right? The physical and physico, that means physical and chemical. So, it is a type of process in which both the physical changes as well as the chemical changes takes place. Okay? And hence it is known as physicochemical reactions. Now, what are the physical and chemical changes that takes place during the photosynthesis? So, the first change is physical change, right? Physical change. So, what type of physical changes that takes place? So, here the exchange of gases takes place, right? Because we know this is the suppose a leaf, and in the leaves, the photosynthesis takes place in the cells, right? So, suppose this is a cell in which there will be CO2 that will be taken from the outside, and then in the presence of water, it synthesizes what? Glucose. The leaf synthesize glucose and its byproduct is O2, right? So the physical change, what physical change is happening here? Here, the exchange of gas. Exchange of gas is taking place. And what gas? CO2 and oxygen. CO2 is taken inside and oxygen is, when synthesized, it is transported outside, right? So the physical change involves exchange of gases, right? And what are the chemical changes that takes place? So the chemical changes, there are a lot of chemical changes such as, but if we see the basic chemical change that is taking place is here the CO2, if you see, we know the formula of glucose, it is C6H12O6. So what we are seeing here is that this carbon dioxide is getting reduced, right? Because here in this carbon dioxide, this addition of hydrogen is taking place and hence it is getting reduced. So we are seeing that photosynthesis is a physicochemical process in which the chemical changes are what? Because here the oxidation and reduction takes place. Oxidation and reduction takes place. oxidation and reduction takes place. And we can see it through this reaction that here CO2 is getting reduced, right? And it is getting converted into glucose. While this water molecule, it is getting oxidized because the hydrogen is getting removed from the water and hence the byproduct oxygen is being formed, right? Hence the chemical changes involve oxidation and reduction, right? Now, what else do we know about the photosynthesis? So it is an anabolic process. Now, what is anabolic? So it is an anabolic process. So we know that what a metabolism is. Metabolism is what? It is a sum total of all the chemical reactions or all the reaction that takes place in our body and that leads to the formation of various products by which our body survives, right? So, but this metabolic process, if you see, so all the chemical reactions that are taking place, the sum total of all this is known as metabolic reactions or metabolism. But this metabolic reaction, it can be of two types. Okay. One is catabolic. Now, what is catabolic? Catabolic is where the large molecules, they break down into smaller molecules. That is suppose the breaking of glucose or breaking of sugar. So that is a catabolic process. While the second type of metabolic reaction is anabolic reactions. Okay. So anabolic means formation of something. Okay. So anabolic process means formation of products. So here there is no breaking down of product, but the formation of new products are taking place. That we can see through the example. Here the carbon dioxide is getting converted into the glucose. The formation is taking place. Hence, photosynthesis is known as anabolic process. Okay. Now, what kind of reaction occur when this glucose formation takes place? So the type of reaction is endothermic reaction. The type of reaction that takes place is endothermic reaction. Now, what is endothermic reaction? So, endothermic reaction means that here the energy gets consumed, right? The exothermic is one where the energy is released, while the endothermic reaction is the one where energy gets consumed. Okay. Now, from where this energy comes so that it can get consumed to form glucose. So this energy for this endothermic reaction, it comes from light. Okay. So, and hence it is known as photosynthesis because light is important for this reaction to take place. Okay. So this is about the basics of photosynthesis, what kind of chemical reactions takes place, or what type of reaction is a photosynthesis. Now, let's talk about the importance of photosynthesis. So the one major importance of photosynthesis is it is a primary source of food on earth, right? It is the primary source of food on earth, right? Because in our food chain, it always starts with what? Plants, right? So plants, and if you see around yourself, the plant is the most abundant, you can say, right? And we know that Rubisco, it will come to it later, but we have already seen in the previous class that Rubisco is one of the most abundant enzymes present, right? And hence, and that Rubisco is involved in photosynthesis, right? And this plant or the photosynthesis is the primary source of food on earth because in the food chain, the plant is getting consumed by the animals, then the animals are getting consumed by the higher animals. But the primary source or the first source is what? It is plant only. And the plant get their food from photosynthesis, right? Hence, this photosynthesis is the primary source of food on earth. Now, second importance is it causes the release of oxygen. And this oxygen is very much important for all the animals on this planet to survive, right? So, and this oxygen comes from where? From the plants. Okay. So these are the two importance of the photosynthesis. That is the primary source of food, and second, it causes the release of oxygen that leads to the survival of all the animals. Okay. Now, let's talk about the requirements. What are the requirements of the photosynthesis? Okay. So, if we draw the chemical reaction, so chemical reaction would look something like this. Carbon dioxide plus water. Now, this carbon dioxide plus water, it forms what? It forms glucose. This is C6H12O6, right? And oxygen. This is a chemical reaction. Now, what does this chemical reaction requires? It requires first light and second chlorophyll. It requires chlorophyll. Now, what is this chlorophyll? It is a pigment, and we will study in detail about this in upcoming slides. Okay. So, if we see what are the requirements, we can see here the requirements are carbon dioxide. What are the requirement? The requirement of photosynthesis carbon dioxide, hydrogen donor. Okay, hydrogen donor. And in this reaction, the hydrogen donor is written as S2O. Okay. Okay. But this hydrogen donor can also be changed. That we'll see. Okay. Now the third is light. Right. And the fourth is chlorophyll. These are the important components that are required for the photosynthesis. If any of these will be absent, photosynthesis won't take place. Okay. Now, what I said that the hydrogen donor here with one of the important component of photosynthesis is hydrogen donor. But this hydrogen donor need not to be water all the time. Okay. And based on this, the photosynthesis is also of two types. Okay. So let's talk about the types of photosynthesis. So this type is based on the type of hydrogen donor. You can say the type of hydrogen donor or based on the byproduct form. Okay, based on the byproduct form. Because why I'm writing on the basis of hydrogen donor or byproduct? Because this hydrogen donor is responsible for the formation of byproduct. So, based on the type of this hydrogen donor, the the byproduct will be formed. The byproduct, what I mean here, the oxygen. Okay, your glucose is your main product, and this is whatever will be formed after that. That is the byproduct. Okay, so the photosynthesis is of two types. So based on this, photosynthesis is of two types. Okay, so what is the first type? The first type is oxygenic photosynthesis. First type is oxygenic photosynthesis. Okay. Now, what is oxygenic photosynthesis? In this type of photosynthesis, H2O or water molecule used as hydrogen donors. Okay. In this type, water molecule is used as hydrogen donor, and hence oxygen is formed as byproduct. Oxygen is formed as byproduct. Okay, oxygen is formed as byproduct. Okay, and hence it is known as oxygenic photosynthesis, or the photosynthesis in which oxygen formation is taking place. Okay. Now, its examples are all plants, and in bacteria, this cyanobacteria, also known as blue-green algae, Okay, so these are the plants and cyanobacteria that leads to the oxygenic photosynthesis. Now, the second type of photosynthesis is anoxygenic photosynthesis. Second type is anoxygenic photosynthesis. Now, what happens in anoxygenic photosynthesis? Here, oxygen does not form as a byproduct. Okay, so in this type of photosynthesis, no oxygen molecule is formed. Why? Because in this case, water is not the hydrogen donor. Okay, so the molecule except water act as hydrogen donors. Okay. Okay, and because of this, the oxygen is not formed, and hence it is known as anoxygenic photosynthesis. And what? Okay, so the example of anoxygenic photosynthesis is purple sulfur bacteria. purple sulfur bacteria. Okay, so this bacteria use what? It use H2S. Okay, it use H2S as a hydrogen donor, and hence sulfur. Sulfur is byproduct. Sulfur is byproduct. So when this purple sulfur bacteria does photosynthesis, it leads to the formation of sulfur as a byproduct instead of oxygen. And hence, this type of photosynthesis is known as N-oxygenic photosynthesis. Okay? Okay. Now, there was one scientist named Julius von Scher. So he performed an experiment, or you can say he proved something. So what we are seeing in the reaction here is that the carbon dioxide is getting converted into glucose, right? But if you see the leaf, okay, and if you look for the glucose in there, you won't find any. Because this glucose, once it forms, it immediately gets converted into another carbohydrate named starch. Okay, so the glucose is never stored in the leaves, but it's get converted into the starch. Okay, so this is what Julius von Scher proved. So what he proved in 1854, he told that glucose formed during photosynthesis. Glucose formed during photosynthesis. What happens to it? It gets converted. Gets converted to what? To starch. It gets converted to starch. Okay. So it will never be stored in the form of glucose. So if you look at the leaves and check for the glucose, you will not find it. Instead, you will find starch. Okay. And he also told that the first visible product of photosynthesis is photosynthesis is what? It's starch. Okay, it is starch. Okay, so if you look into the leaves, the first visible product, although the glucose is formed, it's not visible because it immediately gets converted into starch. And hence, the first visible product is starch. And this is proved by whom? It is proved by Julius von Scher in 1854. Okay. Now what we have seen here, we are talking about the requirements, right? So we saw the requirements are carbon dioxide, hydrogen donor, light, and chlorophyll. But how does people know that these are the things that are absolutely required for the photosynthesis? So this is known because scientists back in 1700, 1800, they did various experiments to prove this thing. Okay. So now let's see what kind of experiments were performed. So the first experiment was variegated leaf experiment. So what is variegated leaf? So before that, let's see one more important thing. We know that the glucose is getting converted into the starch. We saw that Julius von Scher proved this, that glucose gets converted into starch, right? And this starch, it is stored. Okay. It is stored, right? Now, if you want to check the presence of the starch, there is a test by which you can do this. Okay. So a starch can be, if a starch is present somewhere, it can be detected. So starch can be detected by a test. Okay. And the test is known as iodine test. So it is detected by iodine test. Okay. So starch is detected by what? Iodine test, also known as I2 test. Okay. Now what happens? How does starch is detected by the iodine test? So starch in the presence of iodine gives what? Gives blue color. Okay, gives blue color. Even in the potato, there's a large amount of starch. So if you add a few drop of iodine over that potato, after cutting the potato, if you add a few drop of iodine, you will find this blue color. Okay. So starch in the presence of iodine, it gives the blue color. Okay. Now, why I am telling you this? Because this is the way by which scientists, or on the basis of this thing only, the scientists prove that the components that I was telling, that is carbon dioxide, hydrogen donor, light, and chlorophyll, these are important for the photosynthesis. Okay. How? Because they did the iodine test under different conditions. Now let's see what is variegated leaf experiment. So what is, first of all, what is variegated leaf? Okay. What is variegated leaf? So variegated leaf is something that it looks something like this. This is your normal leaf, right? This is a normal leaf, and this leaf is all green, right? But in certain regions, you will find this yellow spots. Okay, you'll find the yellow spots. Okay. Now, if you take a variegated leaf and if you do the iodine test of it, okay, if you do the iodine test of this leaf, what you will find? You will find that this in this yellow region, no color change will be observed. So no color change is observed in yellow region. Okay, in yellow region. This will be your first observation. If you do the iodine test on a variegated leaf. Okay. And secondly, in the greenish part, so these all are the greenish part. So in the greenish part, you will see blue color. So what you will see? You will see blue color in the green portion. Okay. So if you take a variegated leaf, you add a few drops of iodine over it, or if you simply dip this leaf in the iodine solution, you will see two different regions. A region where there will be no color change, and that region will be the yellow region. And second, over the green part of the leaf, you will see a change in the color, and that change is blue color. So what does it indicate? The formation of blue colors, it indicates that starch is present in that region. And if starch is present in that region, it means that the photosynthesis is taking place in that region, right? So the blue color, what it tells? It tells starch is present, and it in turn, what tells? It tells that photosynthesis is happening, right? It tells that photosynthesis is happening in that region. While in the region where there is no color change, it means no photosynthesis is happening in that region. Okay. Okay. So by this, what one can conclude that the photosynthesis takes place in the green region, right? Now, here the scientists also did another set of experiment where what they did, they took a leaf. So this is a plant with the leaf, and then they covered a part of that leaf with a thin black strip. Okay, they covered it with a thin black strip, and then kept it some time in the light for the photosynthesis. Okay, after some time, so they took a leaf and covered the leaf with a black strip, covered a portion of a leaf with a black strip, and kept in light. Okay, kept in light. Now, after some time, they did the iodine test. And what they observed? The first observation. So this is one part. This is one part. So what they observed? They observed that. Okay. So what they observed that the exposed area means this area where there was no black strip. So exposed area give blue color upon iodine test. It gave blue color, while the covered area does not undergo color change. Does not undergo color change. Okay. Now, from here, what they found that if the region is undergoing blue color change, it means that in that region photosynthesis is happening, right? While in the region where there is no color change, means photosynthesis is not happening in that region, right? So from here, what one can conclude that because this was kept in the light, right? So light was falling here, 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 but here because of this black strip, light was not able to reach the leaves underneath it, right? So from this, people concluded that light is important for the photosynthesis. Okay, so through the whole this variegated leaves experiment, what was proved? It was proved that the photosynthesis occurs in green parts. Okay, it occurs in green parts of leaves and requires light, right? Because from the second experiment, this is your second experiment. What we saw that when the leaf was covered with the black strip, no photosynthesis was happening, and the region which was exposed to the light, photosynthesis was happening. Okay, so here what people concluded that the photosynthesis occurs in green parts of the leaves and requires light. Now, what is responsible for this green color of the leaf? So the green color comes from the chlorophyll. So this proves the second point as well, right? Because this chlorophyll is also important for photosynthesis. If there is no chlorophyll present, photosynthesis will not occur. Okay. So this is the one experiment that proved that light and the green part of the leaf is important for the photosynthesis. Now, what was the second experiment? So the second experiment is called as Mole's half-leaf experiment. So this experiment was performed by a scientist named Mole. So the experiment was performed by a scientist named Mole. Okay. Now, what he did and what he proved via his experiment. Let's see. So he took a green plant. Of course, he took a plant. It has certain leaves, right? It has certain leaves. Now, what he did, he dipped a part or a part of a leaf in a solution. Okay. So suppose this is a leaf. So this, he dipped the leaf. He dipped the leaf. Okay. So he dipped, suppose this is the half portion. So he dipped the leaf in a solution. Okay. He dipped the leaf in a solution, half of the leaf only. And this solution is what? This is the CUET solution. CUET solution. Potassium hydroxide solution. Okay. Now, what does this KOH do? This KOH, it absorbs CO2. So KOH, it absorbs carbon dioxide. It absorbs carbon dioxide. Meaning what? Meaning that the leaf that is covered or dipped into the KOH solution, in that leaf, no carbon dioxide will be accessible to that region. Okay. So it means that this KOH, it does not allow, KOH does not allow CO2 to reach leaf, carbon dioxide to reach leaf, right? So this portion will not be exposed to the carbon dioxide. Okay. Now, this is the half. So the half leaf is exposed. So half leaf is dipped in KOH. Okay. And because of this reason, it is also known as moles. It is known as moles half leaf experiment. Okay. Now, after some times, he performed the iodine test. And what he observed, so suppose this is a leaf. This is a leaf. Now, this portion is, say, dipped. This was dipped in the KOH. And this is undipped. This is undipped. Okay. So this portion is dipped in the KOH. And this is undipped portion. And then when he performed this iodine test, he found that in the upper portion, there was no color change. So in the dipped region, there was no color change. While in the undipped region, or the region that was not in the KOH solution, here color changed to blue takes place. Color changed to blue. Okay. So from here, what he concluded that since there is no color change, means there is no starch formation, right? There is no starch formation, means no photosynthesis. Okay, means no photosynthesis. So in the dipped region, or the region that was dipped in the CUET solution, there was no photosynthesis. And why? Because in this region there was no CO2. There was no CO2 available on the leaves, because all the CO2 was absorbed by this CUET solution. So from this, what was proved? From this, it was proved that the photosynthesis requires carbon dioxide, right? Photosynthesis requires carbon dioxide. Understood? So this is the Mohs half-leaf experiment, and it is known as half-leaf experiment because half of the leaf was dipped into the pH solution. Okay. So we have proven that photosynthesis requires light photosynthesis requires carbon dioxide. Now let's see one more experiment, that is inverted jar experiment. So what was inverted jar experiment? So this experiment was performed. This was performed by scientist named Joseph Priestley. It was performed by scientist named Joseph Priestley in the year, in what year? In the year 1770. Okay, in the year 1770. And in the year 1770, the concept of oxygen was not known. Okay, so by this I mean that oxygen was not discovered till then. Okay, so Joseph Priestley performed the inverted jar experiment, in which what he did, he took two airtight jars. Okay, so this is what? This is the airtight jar. By airtight, I mean that no air will be allowed to go inside or outside. Okay, so the air that is already present inside will be inside, and it won't escape. Okay, and also vice versa. With the air from outside, it will not be able to go inside. Okay. Now, what he did, he performed these two sets of experiment. So the first experiment and the second experiment. Now let's talk about the first experiment. So in the first experiment, what happened? He kept the mouse and a burning candle inside the jar. Okay. And he kept it for some time. So after some time, what he observed? He observed that the candle stopped burning, which means candle extinguished. Candle, after some time, the candle extinguished, and mouse died. And mouse died. Okay. So this was his observation after keeping the candle, burning candle, and a mouse in an airtight jar. Okay. After some time. After some time. Okay. Now, from this, what he concluded? So he concluded that due to burning of the candle, due to burning of the candle and mouse breathing, because mouse was breathing there, right? And mouse breathing, due to burning of candle and mouse breathing, the air quality changed. Air quality in the jar changed. Air quality in the jar changed. Okay. So he concluded that something happened due to the burning of the candle and the mouse breathing, due to which the air quality or the air that was present inside this jar, it got changed. And by change, what it means? It means that its quality, or it becomes foul air or bad air, right? And he called this bad air what? He called this bad air as, because at that time oxygen was not known, so he called that air as phlogiston. Phlogiston. He called it as phlogiston. Okay. Now we know that this concept, that it happened because the oxygen that was present in this chamber, it got used up, and hence there was no oxygen present. And people, when we breathe, we release carbon dioxide. Also, this burning candle, it was releasing carbon dioxide. So the oxygen got consumed, and the entire chamber got filled with the carbon dioxide. And because of this, the mouse died, and this candle stopped burning, or it gets extinguished, right? Now, so he performed, this was his observation based on the first experiment. Then he performed the second experiment. in which he took the same setup, but he made one difference or one change, and the change was what? The change was it kept a mint plant inside. Okay, so along with this candle and the mouse, he also kept the mint plant inside this airtight gas as a jar. Then he observed it for some time, and after some time, what he observed? He observed that the candle was still burning. So candle was burning. When he kept this mint plant, he found that the candle was burning and the mice was alive. Mice was alive. So what he concluded from this? He concluded that the quality of air that was changed or that was degraded in the previous case, that is in this case, the quality of air that was degraded, it was somehow restored by this mint plant, right? Because in this case, when the mint plant was there, the candle was burning and mice was alive, right? So this mint plant was somehow it was restoring the quality of air in the jar. Okay, so he concluded that the quality of air was restored due to mint plant, due to the mint plant. Okay, and he called this process. What he called this process is he called the process as dephlogiston. So the process was called as, or the air you can see that it was called as dephlogiston. Okay, we called it as dephlogiston at that time. But now we understood that why this thing was happening, because in the first case, what happened that this carbon dioxide gets accumulated in the jar and oxygen get consumed. Because of this mouse died. And in the second case, this is one mouse, so mouse was alive. And in the second case, this mint plant, because this mint or any plant we know, they produce oxygen, right? So the oxygen was getting used up by the mouse and the burning candle, while this candle and the mouse that release carbon dioxide upon breathing, this carbon dioxide was used by this mint plant. And hence the exchange of gases was taking place, and which kept this mouse and the candle alive, right? Okay, so at that time, because oxygen was not known, so these things were called as phlogiston and dephlogiston. But then Joseph Priestley in 1774, he discovered oxygen. Okay, so in 1774, oxygen was discovered. Oxygen was discovered in 1774. Okay. Okay, so this is the inverted jar. This was the inverted jar experiment, which showed that what the plant does. The plant take carbon dioxide and releases oxygen. Okay. So overall, this conclusion, if you say the conclusion of the experiment was plant takes CO2 and release oxygen. Okay. Now, then came another scientist known as Jane Ingenhouse. Okay. So the Jane Ingenhouse, what he did, he performed this experiment in the similar line as Joseph Priestley, or you can say the experiment of Joseph Priestley. He re-performed this experiment, but with certain modifications. Okay. So Jane Ingenhouse, he in 1779, okay, 1779, performed modified inverted jar experiment. Inverted jar, or you can also see bell jar. This is on bell jar experiment. Okay, so he performed this experiment, but with certain modification. So what was the modification? So he performed this experiment in 2 times, at 2 time points. The first experiment was in the daytime, so perform experiment at 2 time points. Okay, the first experiment was during daytime, and the second was during night time. Okay, during night time. Now we know what will happen at the daytime. So at daytime, when the experiment was performed, both the mouse was alive, and candle candle was burning. Right, candle was burning when it was performed at the daytime in the presence of this mint. Right, but what happened during the night time? So when the similar experiment or same experiment was performed in the night time, you observed what you observed. You observed that mice mouse died. Mouse died, and also this candle is stopped burning and it gets extinguished. Okay, it gets extinguished. So mouse died and candle extinguished during the night time under the same setup. The question is why? So it means that the mint, what conclusion one can draw? So one can say that the mint that was restoring the quality of air during the daytime, it was not able to do the same thing during the night time, right? So the mint was not able to restore air quality during night time. Restore air quality during night time, right? During night time. Now, why it would be happening? What could be the reason? It was because here, what does it mean that photosynthesis was not happening during the night time, right? Why? Because if photosynthesis would have been happening, oxygen formation will take place, right? So it means that oxygen does not form during night by plants. Oxygen is not formed during night by plants. Okay. So if you see what can be the factor that is responsible for this situation, that in the daytime the oxygen is getting formed, while in the nighttime it is not happening. So if you see what will be the difference, so it means that the first conclusion is that photosynthesis does not happen in the night, right? Photosynthesis does not happen in the night. This is the first thing. Fine. But why? Why? Because in the night there is no light, right? There is no sunlight. So it is because there is no sunlight during the night time. And from this, what can be proved? It can be proved that light is important. It can be proved that light is important for photosynthesis. Okay. So via this experiment as well, Jane Ingenhouse told, or he proved, that light is important for photosynthesis. And due to this reason, during night time, no photosynthesis takes place. Okay. Now, he also performed another experiment that is known as inverted funnel experiment. So, what is inverted funnel experiment? So, this experiment is also performed by Ingenhouse. It was also performed by Ingenhouse. Okay. So, what was in this experiment? So, in this experiment, he used an aquatic plant. Okay. So, the plant that he used is aquatic plant, used an aquatic plant. And what was the name of this plant? The name was Hydrilla. Okay. Hydrilla. And what experiment did he do? So, he took the aquatic plant Hydrilla. So, this is a water in which he took this plant Hydrilla and then covered this plant with this funnel. Okay. The inverted funnel. Okay. This is a funnel. And when he exposed this plant with the sunlight, okay, when he exposed this plant with the sunlight, with the sunlight, what he found? He found the formation of bubbles. And by that time, it was known that the carbon dioxide is getting used and oxygen is getting released. So by that time, it was known that something is getting formed. And what he observed that when he exposed this plant, aquatic plant, hydrilla, with the sunlight, he saw the formation of bubbles. Okay, he saw the formation of bubbles. So, which prove that photosynthesis is happening. Okay, which prove that photosynthesis is happening or photosynthesis is taking place. Okay, photosynthesis is taking place, right? So, from this also, where his inverted funnel experiment as well, he proved what he proved. He proved that light is important. Again, he proved that light is required. Light is required for photosynthesis. Okay, light is required for photosynthesis. So what he did, he took a plant named hydrilla, that is an aquatic plant, so it will be under the water. And then what he did, when he used a funnel, and through the funnel he sent that sunlight through that funnel, and he observed that when the plant was exposed to the sunlight, this water bubble formation was taking place. And through this water bubble formation, he showed that yes, the photosynthesis is taking place. Okay, so this was the inverted funnel experiment, and both of this experiment proved what? That light is required for photosynthesis. Okay, now we knew that light is important for photosynthesis, but we also know that light has a wide spectrum. It consists of wavelength of different rays, like of different wavelength, right? So, what was the most important wavelength of the light that is used for the photosynthesis? So, this experiment was done by a scientist named T.W. Eagleman. Okay, so T.W. Eagleman performed the experiment that determined what? Determined which wavelength of light is important for photosynthesis. Okay, so till now we are seeing that light is important for photosynthesis, but what specific wavelength of light is important? It was proven by T.W. Eagleman. So determine which wavelength of light is important for photosynthesis. Okay, so before moving to the experiment, let's see few terms. Okay, so we know what is aerobic bacteria, but just for the revision and just to make a story, I'm telling. So what is aerobic bacteria? Aerobic bacteria are the one that need oxygen. Okay, these bacteria need oxygen for the respiration. Or you can say survival, right? For survival. So aerobic bacteria are the one that needs oxygen for the respiration or survival. Okay, so if you place an aerobic bacteria in certain situation, there is no oxygen, they will not survive there. While or the bacteria will simply move from that region to the region that has oxygen. Okay. Now, the second concept is about the prism. So we also know what is prism. So prism is what? It scatters the light into its components, right? Its components. And what are the components of the light? These are the different wavelength rays, right? So if you have light, suppose this is a prism, okay? So if you pass a light through it, so this is a white light, this is a white light, but we know that this light comprises of its component of different wavelengths. So it will split the light, right? It will split the light in 7 components, right? It will split it into the 7 components, and each of these components have a different wavelength, a specific wavelength, right? So this is the basic. Now let's see what was done by the T.W. Eagleman. So what he did, he used the green elements. to prove that what wavelength of light is important, he used a green algae. He used green algae. Okay. Then named, what was the name of the algae? Name was Cladophora. So the name of the algae was Cladophora. Okay. Now what he did, he kept it in a solution. This is a Cladophora. This is a Cladophora. Okay. In this solution, he also kept the aerobic bacteria. Okay. So the aerobic bacteria were floating in this solution. Okay. So these are what? These are bacteria. These are aerobic bacteria. Okay. Now he took the prism and then passed this white light. Now this white light will be scattered and then it gets and hit the different region of the Cladophora, right? So these are the rays of different wavelength and it reached this Cladophora. So we know this is Vibgyor, Vibgyor. So this is violet to red light rays of different wavelength. Now when he observed it for the some time, what he found? So he found that the bacteria, these aerobic bacteria that were present in the solution, they get accumulated in two regions specifically. One, in the blue region, they get accumulated over this Cladophora in two regions, one on the blue region and the second on the red region. So now you know that when the light is reached to this Cladophora and this is green algae, so it does photosynthesis. It does photosynthesis, right? Now, if it will do photosynthesis, it will cause the release of oxygen. And where there will be a high amount of oxygen, this aerobic bacteria will get accumulated there, right? And on the observation, what they found, that the bacteria gets accumulated in the blue region and the red region. Okay. So here, what I'm trying to say, that higher the photosynthesis, because we know higher the photosynthesis, what will happen? It will lead to the higher production of oxygen. And if there will be higher production of oxygen, there will be increase in the accumulation of aerobic bacteria. It will cause increased accumulation of aerobic bacteria, right? So increased photosynthesis leads to the increased accumulation of aerobic bacteria. So the region where will be higher photosynthesis, there will be higher accumulation of aerobic bacteria. Okay. So he observed that the accumulation was higher in red and blue wavelength region. Right? So it was higher in red and blue wavelength region. And hence, by this he proved that blue and red wavelength, I am representing wavelength by lambda, red wavelength is important for photosynthesis, Okay. So by this, T.W. Eagleman proved that what wavelength of light is important, and the wavelength that is important is what? It is blue and red. It is blue and red wavelength is important for photosynthesis. Okay. Now, so for today, let's keep it till here. In the next class, we will see few more experiments and then we will start studying about the different reactions that is present in photosynthesis. That's all for today. Thank you.